Hello everyone, I am Sagun. I chose to present Portieria hornemannii, which is Sir Patrick's favorite algae and eventually became my fave algae as well. Why? Let's find out. Portieria hornemannii is a species of red algae in the family Rhizophylidaceae, order Gigartinales, class Florideophyceae, subdivision Urodophytina, division Rhodophyta, kingdom Plantae, and domain Eukarya. P. hornemannii is commonly known as the clawed sea moss. This slide presents the maximum likelihood phylogeny of the family Rhizophylidaceae, within which genus Portieria is included. This is lifted from a study aimed to establish the phylogenetic relationships within Rhizophylidacean genera and its close relatives using chloroplast-encoded RBCL and nuclear ribosomal LSU gene sequences. According to phylogenetic analysis, members of Rhizophylidaceae and their close relatives are monophyletic. Family Rhizophylidaceae is most closely related with Calaminia as indicated by the bootstrap and BI support values of 100%. Moreover, Contrarinia is resolved as the sister taxon of a clade uniting Nesophila, Octodes, and Portieria with a high bootstrap and BI values of 99 to 100%. Let's proceed to its general morphology. Portieria hornemannii thallus is typically small, which grows up to 20 centimeters tall. Its color varies from greenish pink to dark red and becomes orange to brown as the specimen ages. Freshly collected specimens exhibit a strong pungent smell. The thallus is composed of erect, overlapping fronts that arise from a small discoid holdfast. One to several axes arise from the base of the thallus. The main axes are compressed to flattened and narrowest at the tips. The thallus is pinnately branched, consisting of up to 5 to 7 orders of alternate or subopposite laterals. The apices are typically incurved, sometimes straight in newly developing axes. Here are sample images of Portieria hornemannii showing the thallus, stipe, blade, and a photo taken in situ. Portieria hornemannii is widely distributed in South Africa and in the Indo-West Pacific Ocean as indicated by the yellow markings. In the Philippines, they can be found in these areas. So you might want to consider visiting these places kung gusto mong makakita ng Portieria in person. Now let's proceed to the question, why do I love this algae? One reason is simply, it looks gorgeous. It features an undeniably captivating bright red pigment and fancy feather-like appearance na parang pwedeng pang display sa Christmas tree. But it's not just about the beauty, of course. Baka iniisip nyo ang babo naman ng rason ni Roel. Portieria hornemannii has so much to offer. It produces a vast range of bioactive compounds that may be exploited for numerous applications. Did you know that Portieria hornemannii exhibits antimicrobial properties? Its extract contains compounds such as tannins, flavonoids, glycosides, phenols, saponins, and terpenoids, which exhibit broad-spectrum antibacterial activity. It inhibited both gram-positive and gram-negative strains, which it has highest inhibition against Staphylococcus aureus. It also inhibited Vibrio parahemolyticus, which is the causative agent of vibriosis in fishes. This algae also contains anti-cancer and anti-tumor compounds due to the presence of polyhalogenated acyclic monoterpene from its extract. This compound exhibited highly differential cytotoxicity against in vitro human tumor cell lines, with brain tumor, renal, and colon tumor cell lines as the most sensitive to the compound. It exhibits antidiabetic properties as well. It showed anti-hyperglycemic activity in chemically induced diabetic rats. The extract decreased blood glucose and showed good signs of regeneration in the cells of the pancreas. P. hornemannii also has anti-deterrent activity due to apacaoctodins A and B, which are effective feeding deterrents toward herbivorous reef fishes. It is a potential source of fragrance for perfumes. It has a distinctive essence of perfume like flower scents and can give off pleasant fragrance due to the presence of this chemical component shown on the slide. This algae is used in the biotransformation of TNT. Microplantlets of P. hornemannii efficiently remove the explosive compound 2,4,6-trinitrotoluene from seawater. TNT is a nitroaromatic explosive that when dissolved in seawater, it becomes toxic to many marine organisms. And lastly, P. hornemannii is exploited for the synthesis of silver nanoparticles. This is a greener and more eco-friendly method of nanoparticle synthesis. The silver nanoparticles were then evaluated for their antifungal properties and revealed zones of inhibition against B. bassiana and M. anisopliae. So generally, I like P. hornemannii because it offers diverse compounds with bioactivity that can be exploited for biotechnological, pharmaceutical, and industrial applications. This concludes my presentation. I hope you have learned something. Maraming salamat and stay safe.